everybody. I'm going to go through some practice questions for the second half of Unit 4 on the digestive set on nutrition as well. So let's see. Here we've got a first question about some digestive processes. And this is one of those select all that apply questions that everybody loves. Which of these are examples of digestion by enzymes? So digestive enzymes are a really important topic in this chapter. And keep in mind, an enzyme, most enzymes are actually protein molecules, so they are themselves big giant organic molecules, but enzymes catalyze chemical reactions. Hopefully you learned about that in Biology 201. And what that means is essentially they are the machines that perform our chemical reactions. So digestive enzymes, their job is to take these big giant molecules that we consume in our foods, our proteins and our carbohydrates and our lipids, mainly. Nucleic acids also are present in the foods that we're eating, but they're not typically considered to be a great source of nutrition like those other things. And the enzymes, their job is to basically chop those up. Remember, those molecules are polymers. They're chains of smaller organic molecules, the monomers or the subunits or the building blocks. And these big molecules in our foods have to be chopped into those smaller ones so that they can be absorbed in the small intestine. So which of these are examples of digestion by enzymes? All right, so how about A, breakdown of starches by salivary amylase? Generally, when you see ASE on the end of a word, that's a pretty good shot. That's an enzyme. Amylase, in fact, is an enzyme that breaks down starch. Starches are polysaccharides. Okay, B, breakdown of proteins by trypsin and chymotrypsin. Oh yes, trypsin and chymotrypsin are enzymes that we call proteases, and their job is to break down proteins into smaller subunits. How about chewing? Yeah, chewing is a digestive process. That's a type of mechanical process of digestion. And so, yes, you are breaking down foods, but on a larger scale. Big chunks of food into smaller pieces of food. You're not down on the molecule scale. Digestive enzymes are working down on the molecule scale. So we're not going to include chewing as an example of digestion by enzymes. Churning in the stomach. No, that's also a mechanical type process. So the churning of the stomach does help on a larger scale break down food particles from bigger chunks into smaller chunks. And you need to do that so that then those digestive enzymes down at the molecule level can have access to those starches and lipids and proteins and break those down into the smaller building block molecules. The movement of small organic molecules across the lining of the small intestine and into the blood, that's a no. That's another digestive process. That's called absorption, which is primarily the job, and you guys should remember this, the small intestine is where most absorption takes place. And then finally, how about the breakdown of lipids by lipase enzymes? Yes, lipases are enzymes that break down lipids. All right, so A, B, and F on those. All right, how about this one? Proteins in foods are digested into these absorbable monomers. Okay, so the building blocks or the monomers of these larger organic molecules, that's something you hopefully learned in Biology 201, but it comes back up here when we're talking about digestion and nutrition, so you got to go back and review that terminology. And you want to do that anyway because it's going to come up in every biology class that you have. All right, so hopefully you're thinking C, amino acids. Amino acids are the smaller organic molecular building blocks of proteins. Sugars, or what's another name for sugar? Monosaccharide. Those are the building blocks of polysaccharides. Carbohydrates, those all fall into the same category. Nucleotides are the building blocks of DNA and RNA, which are nucleic acids. Fatty acids are one of the building blocks that you can find in many types of lipids, mainly your triglycerides. And you also have fatty acids and phospholipids, the lipids that you have in your cell membranes. So go back and review all that terminology. All right, what catalyzes the hydrolysis reactions? And catalyze, if you remember from Biology 201, is an enzyme, what they do is they actually make a chemical reaction more likely to happen. So they kind of speed them up. and force them to happen. That's what catalyze means. And hydrolysis reactions, you may remember from Biology 201, those are breakdown type reactions that involve water. All right, so what's going to catalyze the hydrolysis reactions required for digestion of large organic macromolecules into absorbable monomers? Okay, and we've already talked about that, so hopefully you're thinking digestive enzymes on that one. You should know the other terminology there, segmentation. If you remember, that's something that goes on down in the intestinal tract, where you have alternate sections of the small intestine contract and relax to kind of help move food backwards, back and forth. That also aids with digestion. Peristalsis, that's the molecular squeezing of the digestive tract that propels things from one end to the other. So segmentation is a back and forth movement of the digestive tract. Peristalsis is a one-way movement. And I think you all know what water is, so we're not going to have to define water. All right, which of these is not a digestive process of the mouth? So you want to go back and review the major digestive organs and the digestive processes that take place in them. Here we're just going to do an example question from the mouth. Let's just go through these. A, digestion by enzymes. Yeah, we actually just talked about salivary amylase a little bit. That's an enzyme in your saliva that partially breaks down starches, and so that's gonna start in the mouth as food gets mixed with saliva. Mechanical breakdown, chewing. Yeah, that's a type of mechanical breakdown. Propulsion. Yeah, when you swallow, that's an example of propulsion. You're propelling the food down the esophagus toward the stomach. And absorption of monomers. Hopefully you're picking D on this one. Absorption primarily occurs in the small intestine. That's the organ you mainly want to associate with absorption. You don't have absorption of nutrients going on in your mouth to any large scale. And that's mostly, a key reason for that is because most of the foods that you take in are not broken down enough. They haven't been, you got these big molecules and just chewing and a little bit of action of salivary lipase. You've also, salivary amylase, I'm sorry. You've also got a lingual lipase. Lipases break down lipids. Lingual refers to the tongue, so it's actually secreted by the tongue, and it helps digest some of the fats in our diet. All right, which of these is not produced by the stomach? So go back and review what is produced by these different digestive organs. Let's just go through them. Pepsin. Yeah, pepsin is that's a protease. It's an enzyme that breaks down proteins. Remember, the stomach first makes it as pepsinogen. That's an inactive form. And then the HCl, that's also made by the stomach. That's your hydrochloric acid that's released out of those gastric pits. That converts pepsinogen, and you guys have seen the inogen term multiple times this semester. Inogen tells you it's an inactive form. The hydrochloric acid converts it to pepsin, and now pepsin can start partially digesting the proteins that are passing through your stomach. It's not going to break them all the way down into individual amino acids. That's not going to happen until you get to the small intestine. But that process gets underway in the stomach. Amylase, that's actually not something we associate with the stomach, so we're going to X that out. Amylases, again, are enzymes that break down starches. Chyme, yeah, chyme is the name for the combination of food material and gastric juices when all that gets wadded up together in the stomach. And then it's going to move from there through the pyloric sphincter into the duodenum. That's chyme. Intrinsic factor, remember that? That is actually produced by cells along the stomach lining. And do you remember from going way back to the beginning of the course what that is, or not quite all the way back to the beginning, but in Unit 2, intrinsic factor is made by the stomach, and it is necessary for you to be able to absorb vitamin B12 from along your intestinal lining. So if the stomach is not producing intrinsic factor properly, you can't absorb vitamin B12 adequately through the wall of the small intestine, and you need vitamin B12 for cell division. So it has a big impact on your bone marrow where your blood cells are made, and so people who have this problem can wind up with a type of anemia called pernicious anemia. A little bit of review from Unit 2 there. Okay, another question about the stomach. Proteins are completely digested into absorbable amino acids in the stomach. And hopefully, based on what we just said, you're thinking false on that one. They are partially digested by the pepsin. But if you think about proteins as, oops, hang on. Didn't have my pen turned on. All right, if you think about proteins as being chains of amino acids, and of course they really have a 3D shape. In the stomach, you might break this down with pepsin. Pepsin breaks it down into shorter chains of amino acids. And then as these guys, they're going to move through the pyloric sphincter down into the duodenum and the ileum, and in there you're going to have additional proteases from the pancreas. You've got the trypsin and the chymotrypsin. That's going to chop these chains of amino acids into even smaller chains. And then finally you have enzymes along they call them the brush border enzymes that are sitting along the membrane of the intestine lining cells itself, the cells that line the small intestine. And those enzymes finally chop these things up into individual amino acids and allow you to absorb them through the lining. All right, moving on to the small intestine here. Which of these increase surface area in the small intestine? We want to select all that apply here. Remember, surface area is very important for what? What function of the digestive system is increased if you have more surface area to do it on? Hopefully you're thinking absorption is really key here. If you have more surface area of the intestinal lining facing the lumen, the interior space, that's going to give you more surface area to absorb nutrients. With less surface area, more of these nutrients are just going to pass unabsorbed through the small intestine, and then once they get down deeper in the small intestine and on into the large intestine, they can't be absorbed very well anymore. All right, the hepatopancreatic sphincter. No, hopefully that doesn't really have anything to do with surface area. That is a sphincter, a little tiny ring type muscle that opens and closes the opening that's allowing bile to come in from the common bile duct and all those wonderful pancreatic enzymes that are coming in from the pancreatic duct. So that's not really involved in, certainly very important for digestion, but it's not involved in surface area. Circular folds, yes, those are ring-like structures you have along the inside lining of the small intestine that increase the surface area, and those are on a larger scale, like you can see it with the naked eye type scale. The cecum, no, the cecum is the first part of the large intestine. Villi. All right, so circular folds are at more of kind of a naked eye scale, and then as you zoom in closer and closer to the lining, you'll see that along those circular folds, the lining is kind of arranged like that. Those are called villi. Go back and look in your textbook at the pictures. Microvilli, so if you zoomed in on a single cell along the lining, and up here the part of the cell that faces the lumen right there, the cell membrane has lots of little tiny microscopic villi or hair-like projections, and those are called microvilli, and then some people call those part of the brush border. And along the microvilli, you've got membrane proteins that absorb nutrients, and you've also got enzymes sitting along the microvilli that help complete the digestive reactions so that you wind up with absorbable monomers. All right, a little bit about, go back and review what we discussed about the small intestine hormones, the endocrine hormones of the digestive system. Which small intestine hormone stimulates secretion of bicarbonate-rich pancreatic juice? And hopefully you are picking secretin. So secretin is a hormone that is produced by the small intestine, and probably its major function is it gets into the blood, but its target cells are actually within the pancreas. So it stimulates cells within the pancreas to secrete their bicarbonate-rich pancreatic juice. The same bicarbonate we learned about in the respiratory system, and bicarbonate ions are alkaline. They're alkaline compounds. And so what are they going to do to pH? They raise the pH. So why does the pancreas, these are actually made by the pancreas, the bicarbonate ions, or they're secreted into the pancreatic juice by the pancreas, which makes sense. Why do we have those? So think about what's coming in from the stomach. That chyme coming in from the stomach has lots of hydrochloric acid in it, which has a very low pH. The bicarbonate ions are in the pancreatic juice to neutralize that. So you don't want the chyme to retain or keep an acidic pH when it comes into the duodenum, because that would be damaging to the inside lining of the small intestine. The cells there can't tolerate that low pH. So the bicarbonate-rich pancreatic juice helps neutralize or raise the pH back towards a neutral level. Gastrin is a hormone produced by the stomach, which increases the activity of those digestive glands in the stomach down in the gastric pits. And trypsin and pepsin are not hormones. They almost sound like they might have hormone-like names, but they are actually, both of those are protease enzymes. All right, how about this one? Which small intestine hormone stimulates secretion of enzyme-rich pancreatic juice? So the last hormone we talked about stimulates the pancreas to secrete all these bicarbonate ions. What hormone stimulates the pancreas to release a bunch of enzymes? And it also stimulates the gallbladder to contract. And what do we have stored in the gallbladder? Hopefully you're thinking bile. All right, so this enzyme tells the pancreas, release all these enzymes for digestion, and it's also telling the gallbladder to contract, which is going to squeeze bile out of the gallbladder through the cystic duct, down into the bile duct or common bile duct, and then that's going to pass through the hepatopancreatic sphincter, the opening there, and into the duodenum. And what does bile do? We say bile emulsifies fats or lipids. Kind of functions like a detergent. So it breaks apart balls of grease or lipids that are entering the duodenum from the stomach and makes them more accessible to lipase enzymes. The lipase enzymes can actually get in there and break apart those triglyceride molecules that are coming in from the stomach from the foods that you've eaten. All right, so on this one, hopefully you are thinking about CCK, which stands for cholecystokinin. So that hormone, so there actually are cells in the small intestine. It's a small intestine hormone, and as chyme starts moving in from the stomach into the duodenum, these cells then release CCK, and CCK is endocrine hormone, gets into the bloodstream. Its target cells are in the pancreas and in the gallbladder, and results in those actions that we talked about. All right, which of these is the only digestive function of the liver? Certainly not the only function of the liver, but the only digestive function. All right, and remember, production of bile is a key function of the liver. So you've got cells in the liver that produce the bile, and that bile gets released into little tiny bile canaliculi, little canals, and then ultimately Those enter into the hepatic bile ducts that carry the bile on down towards the common bile duct. But if you don't really need to release the bile, then it backs up through the cystic duct over to the gallbladder for storage. So that's a major digestive food breakdown function of the liver. Now, if you go back and review what we learned about the liver does a lot of processing of the nutrients that get absorbed by your small intestine. So remember, we learned about the hepatic portal circulation in Unit 3. So some of that should start to make more sense now that you've studied the digestive system. The blood that is draining off of the digestive organs, like the wall of your small intestine, ends up in, say, the superior mesenteric vein, which then connects to the hepatic portal vein. So that blood is going to be full of all these nutrients you've absorbed from the food that you've eaten. And so that gets carried off to the liver, and then the liver, you've got all these hepatocytes, these liver cells that process a lot of those nutrients. The liver processes a lot of lipids. The liver will absorb sugars and store them as glycogen or convert them to fats, things like that. So the liver certainly has a lot of processing of nutrients that it does. But in terms of actual digestive function, taking bigger molecules and turning them into smaller absorbable ones, bile production is the only liver function. All right, liver and gallbladder. If you had a pancreatic tumor that compressed the common bile duct, what would you expect? And this is actually if you've ever known somebody who's had pancreatic cancer. Unfortunately, it's not all that. Most of us probably do know somebody who's had that. And a lot of times the tumor is over on the part of the pancreas close to the common bile duct. And so if it compresses that common bile duct, you're like over here on the side of the liver, here's your common bile duct coming down, you get a tumor growing here, and it squeezes the common bile duct. The common bile duct connects to the cystic duct. There's your gallbladder, and then goes on up here into the liver. Guess what? All that bile backs up into the gallbladder and up here in the liver. And remember, bile really gets part of its name from bilirubin, that breakdown product of heme that we learned about back when we studied the blood in Unit 2. So if the liver is not able to keep producing and secreting bile because you have this backup, then those liver cells don't absorb bilirubin out of the blood properly anymore because it doesn't need to, because it can't keep making this bile that's just going to back up. And then that bilirubin gets deposited in your tissues and you wind up with jaundice. So hopefully you were thinking A there for jaundice. If we had another answer here, E, problems with digestion of lipids, that would have been a correct answer as well, because if the bile can't get into the duodenum, because you've got a pancreatic tumor that's compressing the common bile duct and blocking that from happening, then you can't digest your lipids as well. And that's probably going to lead to a diarrhea type problem. Lots of digestive problems, and diarrhea would then be likely because you can't break down these lipids and absorb them properly. All right, pancreas, which of these is not contained in pancreatic juice? So go back and review, what, oops, hang on, sorry. The pancreas makes lots of things that are important for aiding food digestion. Most of your digestive enzymes are made by the pancreas. That doesn't mean that food enters your pancreas for digestion. It's staying in the small intestine, but the pancreas makes a lot of digestive enzymes that get squirted through the hepatopancreatic sphincter into the duodenum, and then they move down from there into the jejunum. And they're very important for digestive processes. All right, so do we have alkaline compounds? Oh, yes. We just talked about that. Lipases, yeah, the pancreas does make some lipase enzymes that help break down lipids. Remember, the lipases won't work very well unless you have bile around. Bile helps break down those big fat globules down into smaller ones so that these lipase enzymes can actually get to the lipids and break them down. Water, oh yeah, you've got any body fluid is going to have water in it. Active proteases, that's a no. The pancreas does make proteases, but remember they are in an inactive form, kind of like we were talking about with pepsinogen in the stomach. And then after they enter the duodenum, they become activated. And go back and review what we learned about with that, with regard to that. Well, let's see. This thing's not behaving. Go away. Answer there is D. All right, here's a true false. Blockage of the pancreatic duct would not likely interfere much with protein digestion since most proteins are digested completely in the stomach. We actually already covered that concept. That's going to be false. Most of the proteins coming in from the stomach are partially broken down by pepsin, but you got to have these pancreatic proteases like trypsin and chymotrypsin and carboxypeptidase to chop them down into even smaller chains of amino acids. So a big protein molecule is a long chain of amino acids. Pepsin converts it into shorter chains. Trypsin, chymotrypsin, carboxypeptidase from the pancreas converts them to even shorter chains. And then finally, you've got little brush border or microvilli enzymes sitting along the little microvilli of the cells that line the lumen of the small intestine itself that finally chop them up all the way to individual amino acids. All right, the final conversion of organic nutrients to absorbable monomers generally occurs through the actions of what? I just kind of gave that one away. Hopefully you're thinking brush border enzymes. And again, go back and look at your diagrams in the digestive system chapter and make sure you're understanding what is meant by a brush border enzyme. The brush border is another name for the microvilli along the surface of the cells that line the inside lining of the small intestine, and you actually have enzymes sitting there in the membrane that do the final chemical reactions to chop things like short chains of amino acids down into individual amino acids or disaccharides into monosaccharides, things like that, so that you can actually absorb those into those cells and then they get transferred into the blood. Okay, here we go. I think I just gave away this one too. Sugars and amino acids absorbed by cells lining the small intestine are transferred into what? So they travel across the cells, and then so they're absorbed over on the brush border side. That's their microvilli side. That's facing the lumen of the small intestine. They cross the cell, and down here on the other side, that's facing the wall of the small intestine itself, and inside there you've got lots of capillaries around to absorb those nutrients. So that's how the nutrients get from the inside of the small intestine and into the bloodstream. So our answer there is going to be capillaries. Now remember, go back and review how it's different though for lipids, hydrophobic nutrients. They get packaged up in these little lipoprotein particles called chylomicrons. So those are combinations of proteins and lipids, because lipids can't just float around in your body fluids because they're hydrophobic. They hate water. So they get packaged up in these little protein lipid balls, basically, and they're called chylomicrons. And those get sent into the lymphatic system. They go into the lacteals, which transport those through the lymphatic system first, and then eventually they go to the liver for processing. And then eventually they wind up in these other types of lipoprotein particles for transport through the blood, the VLDLs and the LDLs and the HDLs, which that comes a little bit later. All right, kind of a tricky question here. Bile cleaves triglycerides into glycerol plus fatty acids. So a triglyceride is a type of lipid, and it consists of a molecule here called glycerol, and then you've got 3 fatty acids. Okay, so the question is asking us where a lot of the dietary fats that we take in are in the triglyceride form. And you may have heard of triglycerides before. If you've ever had blood work done, you might have had your triglyceride levels checked. All right, so does bile break a molecule like this apart so that the glycerol is separated from those 3 fatty acids? Might sound reasonable at first, but that's actually false, because remember, the job of bile is to emulsify or to help dissolve these larger fat globules into smaller fat particles, and then it's the lipase enzymes. Those enzymes are what actually break individual triglyceride molecules apart. So you're moving from a larger scale down to a smaller scale when you're comparing and contrasting what bile and these lipase type enzymes do with your triglycerides. All right, another intestine question. Although digestion is completed in the small intestine, most absorption of nutrients really occurs in the colon. All right, so sometimes those true-false questions can sound pretty reasonable. You gotta know your stuff. So as I've been saying earlier, almost all absorption takes place in the small intestine. And y'all, please do not make the mistake of thinking that almost all of your digestive tract is called the stomach. The stomach is a very distinct organ which has its own functions. A lot of what we think about as typical digestive activity that we might say, oh, this is going on in my stomach, it's really happening in the small intestine. Even when you have like a stomach ache, a lot of times that's really a small intestine ache. So be sure, since you're going through an anatomy and physiology class, you use the correct anatomical terminology. All right, so most absorption actually happens in the small intestine, so this one is going to be false. You do have some absorption that takes place in the colon, especially of water that didn't get absorbed in the small intestine. You want to make sure you absorb as much of that as possible. And so that gets finished up in the colon. Boy, I'm really good at giving these next questions away before we get to them. Most water is absorbed by what? All right, you have to be careful with this one too, because when you're reviewing, you might get used to, okay, the colon absorbs water, but actually most of it gets absorbed by the small intestine. Any leftover water, though, gets picked up by the colon. Remember, the colon is finishing off the formation of the feces. Not pleasant to think about, but if the colon was not absorbing that residual water, you might wind up with diarrhea type feces more often than you would like. The colon is also involved in absorbing some vitamins as well. The bacteria that live in your colon, along the lining of the colon, you got tons and tons of bacteria there, and so many of them produce vitamins for us that we can't make. They also help break down some foods that we can't break down ourselves with our own digestive enzymes, so they do help out with some of those things. So you do have a little bit of absorption going on in the colon of nutrients, but not nearly as much as what happens in the small intestine. All right, a little bit more about blood lipid. This gets into some terminology that we hear a lot about in healthcare and our own personal health as we get older. How are cholesterol and triglycerides transported in the blood? All right, cholesterol and triglycerides both fall into what organic molecule category? Hopefully you're thinking lipids. And then what's special about lipids? They are, hopefully you remember from earlier classes, they are hydrophobic, so they repel water. They don't like water. So they can't just float around in the watery-based plasma of our blood. They've got to be packaged up with proteins in complexes that help them travel around in the blood. All right, and so because of that, our correct answer there will be D, within lipoprotein or lipoprotein particles. This iPad's driving me crazy. Come on. All right, which type of lipoprotein particle is generally considered to be the bad cholesterol, since excessive amounts can be unhealthy? This is something you should know. And even as a person who's never had an A&P class, you've no doubt probably heard some of this terminology, especially if you're over 35 or 40, or if you talk to relatives who have these types of tests run. The bad cholesterol particles are the LDL particles, which stands for low density lipoproteins. And the LDL particles are generally transporting various kinds of fats, not just cholesterol. And it's called the bad cholesterol, but you don't just have cholesterol in there. You've got various types of lipids, triglycerides, that are being transported in these little particles from the liver to your tissues. So they're supplying these things to your tissues because they actually need them. The problem comes in if you have a diet that causes you to produce too many of these LDLs. It's causing the liver to send too many of these lipids and cholesterol packages out to the cells and tissues of your body. If you have more of those than you need, more than your tissues can actually absorb to use them, then they tend to stick to the walls of your arteries, and they're responsible for causing a lot of the plaque buildup in the walls of our arteries. And that just gets worse and worse and worse as we get older. The American diet is very bad about sending your LDL levels too high, and those LDL particles love to get embedded in the walls of your arteries and trigger that whole process of arteriosclerosis that we learned about back in Unit 3. All right. Answer there was B. Let's move on. Okay. Which type of dietary fat increases LDLs but also decreases HDLs? All right, so if you go back and review what we learned about that, the answer there is A. Remember that HDLs are sometimes considered to be your friendly cholesterol particles. And again, it's unfortunately, these things are referred to as cholesterol, but they're really not. They're combinations of lipids and proteins. Yes, cholesterol molecules are in them, but cholesterol molecules are a certain type of lipid. You have other lipids in there as well. And the HDLs generally are transporting these substances back to the liver so they can be reabsorbed by the liver. Or they're sometimes said to scrub our tissues of excess lipids. So the HDL are generally considered to be a good thing because they're carrying the excess fats away from your tissues and out of the bloodstream toward the liver, which lowers the amount that will build up in the walls of our arteries and generate all that nasty plaque, which incidentally, plaque is, I saw plaque on a personal level for the first time over this past summer of 2014, when I got to do a cadaver dissection class, and it's actually crunchy. So if you ever want an incentive to change your diet, seeing what plaque buildup looks like in the walls of the arteries of a cadaver is pretty eye-opening. It's pretty gross. All right, so it's a certain type of dietary fat that increases LDLs but decreases HDLs. So you should know both of those things are bad. High levels of LDLs, that's not a good thing. And then on top of that, to have low HDL levels, that makes things even worse. Both of those things make it more likely that you're going to develop cardiovascular disease, where you have the deposits of these plaques in the walls of your arteries. And the answer on that one is the trans fats. We've heard a lot about those have really been in the news the last several years. Trans fats are actually artificially created fats uh, that do these horrible things to our lipoprotein profiles in the blood. And because of that, they've got a really bad reputation, and a lot of them have actually been removed from our foods. Food manufacturers have found other types of fats that probably aren't all that healthy either, but not as bad as the trans fats. You guys might remember several years ago, it was real common for people to buy and use Crisco to fry chicken. Crisco is basically just a bunch of trans fats, and so that's, things like that are not in as widespread use anymore, and they've changed the recipes for a lot of those things so they don't contain as many trans fats. On the flip side, your omega-3 polyunsaturated fats, that's a type of what we call an essential fatty acid, and which means our body can't manufacture those on its own, and we've got to have them for proper cell function. And so we're encouraged today to eat a diet that's rich in omega-3 polyunsaturated fatty acids. It's hard to do, though, because they're not present in a lot of modern foods. You find them in fatty fish, for example. That's probably the best source of them, and most of us don't eat a lot of fatty fish in our diets. A lot of people take fish oil supplements. That's why, it's to get those omega-3 polyunsaturated fatty acids, the levels of those up in their diets. Okay, those were just some example questions on the digestive system. And of course, use the review activities posted on Blackboard and go back to your mastering homework for additional examples. And if you have any other questions, please post them for us on the discussion board. Good luck.